Good morning. I want to tell you something about VLIZ blooms in the Belgian coastal zone and how the Cytosense flowcytometer can be used to monitor them. So, phytoplankton are the base of the marine food chain, and they are responsible for roughly 50% of the global primary production. But there are also some harmful algae species which can produce toxins, produce anoxic conditions, and block available sunlight for other species. One of these harmful algal species that is present in the Belgian coastal zone is Phaeocystis. You see a picture of a colony in the middle. The Phaeocystis can obtain high densities during a bloom, and at the end of the bloom, the foam-like structures of the colonies wash into the beach, which you see on the other two pictures. So it's important to monitor phytoplankton and especially harmful algae. So how do we do that? One of the methods in the Belgian coastal zone is by using the Cytosense flowcytometer. It's installed on the research vessel the Simon Stevin and connected to an underway system which pumps water from 3 meter depth, and the flowcytometer analyzes that water for the phytoplankton community. So it can do that every time the Simon Stevin sails for a scientific campaign. We develop different measurement protocols so that we can analyze the complete spectrum of the phytoplankton. And abiotic parameters are monitored in parallel. So what is a Cytosense flowcytometer? It's a scanning flowcytometer, which means that when the phytoplankton cell passes the laser, the complete signal is recorded, resulting in a scan profile, which you see here on the right. What's nice on our Cytosense is that it also has a camera module, so you can link the pictures that are taken with the scan profile. So from the scan profile, 55 different parameters are deducted. Then you have to think about the sample size, the size of a phytoplankton cell, or the fluorescence red, which can be used as a proxy for biomass. And the clustering of the phytoplankton species is based on these parameters and on the photos. So what you see here are all measurements taken with one protocol. You see that there's a large spatial difference in where the measurements are taken. This is especially clear if you look on a smaller time scale. These are the measurements of week 8. These are the measurements in week 13. So to overcome that, I created a grouping layer so that I can group measurements and spatially and temporarily analyze the measurements. So now I'm going to show some results. What you see here in the blocks are the average total fluorescence rate, which was a proxy for biomass. And what you see here are the spring diatom bloom. It starts in the southwest and it moves slowly to the northeast. And after the spring diatom bloom, when all bioavailable silica is taken up, Phaeocystis starts blooming. And you see that it starts, the onset is in week 14, and that it's reached its highest density in week 18. And what was also nice that our data showed that was a small peak of Phaeocystis in the northeast in the autumn in week 37. So that's visible here. So short conclusions. The Cytosense flowcytometer can measure phytoplankton on a high spatial and temporal resolution. The data from 2016 show the spring diatom bloom and the subsequent Phaeocystis bloom, which is characteristic for the Belgian coastal zone. But also, our data also showed a small Phaeocystis bloom in the northeast in autumn. Some future perspective with this data set is create a long-term database for the phytoplankton community composition and identify key parameters for phytoplankton blooms. Thank you for your attention. These are my contacts.